much. Thank you, the organizers, for the invitation. So today I will not talk so much about selection mutation, at least not until the perspectives, I think, but we'll discuss to how establish links, formal links between these two worlds that are the microscopic and the macroscopic scales. So in the recent years, biologists now have the tools to actually access these two different worlds. And it's not evident how to establish a relationship between the two of them. So we have microscopic data, we have also population data, and sometimes the results do not communicate, sometimes they're not the same, and it's not a trivial thing to know why do we have these differences or we don't have them. So the idea is to establish this link. In particular, we're going to work with bacterial data. So we're going to look into Escherichia coli, which is a bacteria that's almost everywhere, particularly in the human being. And it's really the model for microbiologists to study. And we have access to tracking data, so we are able to follow single bacteria for a very, very long time. I will show you how next. And on the other side, we have the possibility, so that we have before as well, to take a picture of the given population at a certain time. And the idea is to relate those two data. So essentially, when the microbiologists will take a picture of the bacterial data, they wouldn't do it at a random time, but they will wait a long time until we'll have a sort of a steady state population. So we'll have a growing population, so it's not a steady state in the sense that the number of bacteria will always grow, but we hope that the structure below, so the age structure or the size structure that we're going to see now, will remain stable. So from a mathematical perspective, we want to see, we'll construct an individual-based model for the microscopic scale and try to relate that to the integral differential models and microscopic models that we see when we formulate the population models. So we're going to go in the direction of the green arrow, so what we call the direct problem. So we will pass from the microscopic data to the microscopic one. There are also some works dealing with the inverse problem. So we observe the distribution, for example, of the sizes of the bacteria at a given time, so at the steady state, for example, and we hope to retrieve the individual parameters, so the division rate, the growth rate, and so on. So there are a lot of work on that, in particular from Marie Doumic, Benoît Pertam, others, that deal with that. So it's a severely ill posed problem. We don't really have a lot of information in the inverse problem, so we're not going to talk about that today. So first of all, for the microscopic data, I would like to thank, so this is all original data that was acquired in Myriam El Karoui's lab. It's one of my thesis advisors, by Sébastien Jaramillo and James Broughton. What we have seen now, it's really wonderful data, and we have access to really these different scales. So to acquire what happens in the single cell level, we'll use what we call a mother machine. So we have different channels, and we capture only one cell in each one of these channels. So we're able to follow them because they are trapped for a very, very long time. If you try to track bacteria like that in an essay tube or in a petri dish, it would be really hard to follow them for a very long time because they're going to overlap. And so in this kind of setting, we actually can access to the steady state information and follow that through time. So if you concentrate in just one channel, for example, and takes pictures of this very channel in some very different times, we'll observe something like that. So we have the same channel through time, sorry, here. So I have the same through time, and we have this kind of cut genealogical tree. So we do not have the whole genealogical tree because we have this channel has a limiting size. So when bacteria reach it, they live. So we do not have complete information about the genealogy, and there's some really interesting statistical aspect about the bias that we can find there. For now, we're not going to deal too much with that, and we want to just concentrate in this, so the lineage that is in top, and which we have full information about. So if you look at that, you have the sizes that we're going to pick like that, and then what I mark there in red is the division times. So we have that. Here we have 12 cells. We have really hundreds and hundreds of cells, and for different media, so growing media. And the idea is to try to compare that and use that information to simulate first, and also to try to have a mathematical comprehension of what happens in the population using those estimates. So for example, if using that data, you can look at the birth size of each cell, so between two red bars, and we have, for instance, the distribution like that. So for different media, you have the distributions of the birth size, and the idea is to use these individual-based distributions to calibrate a model that will predict the population. So these distributions are really different that we'll observe in a population. In a population, you won't observe, for example, the birth size, so it's like a hidden variable. We'll observe just the current size or any other current visible variable. So the idea is to use that to estimate, in our case, the distribution of the current size we observe in a population at the steady state. So to do that, we have to first construct a meaningful mathematical model. So I already said, an stochastic individual-based model. So first, we look at the division times, and we see that there's no surprise, they are not exponential. So we don't have a Markovian behavior in the division times. So the idea is to include an age structure that is going to explain that, and is going to help to establish a Markovian model. In particular, in the recent years, there's a particular age structure that has been discussed. I had a figure, but so is the other model. So we have my bacteria here at some time, then it will grow until this distance here, and this added distance here. So the difference between, the difference between my size at time 0 and my size at time t, it will be my age structure. So it's, since it's the added size, it will grow also at an exponential rate, if my growth is exponential as we saw before. And it also has this not really nice degenerate structure in the sense that the age is not independent of the size. So the age will grow actually at a rate that is given by the size. We're going to see that as well. And we will have this codependency between the two structuring variables. So if we see that in the data, for example, so here each point is a bacteria, is a bacterium. So sorry, it says time there, but it's the added size. So we have the added size and the current size in the ordinates. And we want to see, so this two-dimensional structure. We'll also suppose, and this is the other model hypothesis, that the division time, so each time I have a dot that is going to come back to 0, is decided only as a function of the added size. So we'll want to fix actually the law of the added size, and we want to build a model such that this law is independent of any other variable. So the added size at division is in particular independent of the birth size, for example. So to do that, so first we establish our individual base model. So we write that as a discrete measure. So each dot that you saw before, we want to put that as a delta, as a Dirac delta. And we'll construct our process in such a way that is solution of, so it can decompose as this semi-martingale. So in the martingale here, which is a square integral vertical, you can see it as the variance, and the mean behavior is what's happening here, so which is given by this generator Q. And we see first this first term, which is the growth. So we're going to suppose which is based by earlier biological work that we have an exponential growth, which depends and will, for simplicity, will be the same for every member of the population at rate lambda, which is my elongation rate. And in the second term here, you have the division. And you see that, so at the division, I will replace my cell, my mother cell, by two daughters, with the sizes are given by this kernel F, which is, so it's a distribution in 0, 1, which will give the ratio between the two daughters. And the division rate, what you observe here, you see that is actually the growth rate, so lambda times the current size, time a function b, which depends only on the age, so in the added size. We have that appears because by construction, I told you that I wanted to fix the law of the added size at division. So if I said, for instance, that I want that my distribution function of the added size at division follows this sort of exponential generalization. So I can interpret that. Yeah.
Could you point at the screen because we can't see the mouse?
Okay. Yeah. Sorry for that for the people in Zoom. I can see it there. Yeah. So I was telling you, so the distribution of the added size at division is given by that. So we can interpret this b function here as a rate. But be careful because this b here is not, so it's not in units 1 over time, but in units 1 over length. So it's not an actual rate. And that's the reason because why we have this lambda y here, because we have to multiply this dv, so this sort of division rate that is in units 1 over length times the growth rate to have our actual division rate, so in units 1 over time. So that's our model. So we have our stochastic particular model. So from that, to use a distribution for the Madrid machine to fit that is pretty much straightforward because we have already, since I said that I want to fix my distribution of the added size at division, I just had to look at them and try to fit them and find the b that it's the best. So we first tried some parametric setting. So for a different family of models, we choose the log normal distribution to fit that, so we can fit the distribution of the added size obtained by the Madrid machine and to obtain the parameters and then to infer the form of b using them for the 3 different media. So this is the form of b for a log normal model. So you have that for the 3 different media, and we can already say something about the different media. So for example, if you look, we have glycerol, glucose, and glucose plus amino acids. So glycerol is the purest one. So bacteria grow really slowly, and they don't have many nutrients to reach a very long size. So we'll have here, sorry, I don't see the mouse. Yeah. So we have here a very high peak. So we have a really strong division rate, so that won't allow bacteria to grow too much. That won't allow bacteria to add too much size. In the other side, for example, for glucose and glucose plus amino acid, where bacteria have already more nutrients, they can reach bigger sizes, so the control is weaker. For the function f, so for the ratio between the mother and daughter, is straightforward as well. We use a beta distribution. It's not perfect, but it gives the a nice beginning to use that now to simulate now our population based on this the stochastic individual model. So we have the data from the Madrid machine. We estimate our distribution. We can simulate the process and then to compare with the observation that we have in the population data. We can also do that, so that's from a numerical point of view, try to do that from a mathematical point of view. So because we know that what we observe in the population is the steady state, or we hope that it's the steady state. So what happens when we try to observe the steady state of us a stochastic process. So we'll look at the expectation of a stochastic process, so given by the first order semigroup, so M of t, which if you come back to the semi-martingale is associated to the generator Q, so which consisted in the growth and the jumps at division. The thing is, it's not a Markovian semigroup because the population grows exponentially. So there's some steps to do before to be able to conclude that we have exponential convergence. So we have a unique stationary measure, and we will converge to it at exponential speed. So these kind of results can be obtained using very different techniques, from an spectral point of view, using more PDE-based techniques. Here we are going to use a more probabilistic approach based on Henri's theorem that I'm going to show you next, that allows to have a little more insight in what happens in the convergence. So, and we also, so this is the shape of my stationary distribution, which is this P function here, was already known since the 90s from models used to model plants, with also really, fairly recently re-derived with the exponential convergence using PDE techniques by Hugo Martin and Pierre Gabriel. And we'll give here an alternative approach that we'll see that also can be generalized to other models that are similar to this, in this sense that we have two variables and a degenerate dependency between the two of them. So Henri's theorem, we need mainly two criteria to prove this exponential convergence. So this is one of the earlier versions of this theorem. There are also more modern ones by Herrera, Mattingly, Canales, Migler in the. So this is in the conservative setting. So when I have a Markovian semigroup, we also have similar theorems in the non-conservative case, proved very recently by Vincent, Cloez, Gabriel, and Ali Marguet, and with a twin paper by Cloez and Gabriel, who gives more tractable conditions to prove that. We'll use that version when we require what we call a petit set condition. So we know, will we try that for every compact set. So I have my space here of added size and current size. So I only move in this semi-space here. And I want that for any compact space, so K. The idea is that for any point here, I can reach in some time any other point. And if I'm outside, there's some function, so this V function, this Lyapunov function, that will draw me to this compact set. So this V function is sort of an energy potential that will drive me to the compact set where I know that can explore well the space. So this exploration of the space in my compact, is given by that. So this what we call the Dublin minoration condition, and we also need this Forster-Lyapunov drift condition that will help me to restrain my explorations. And if we have both, we can prove the exponential convergence towards the unique stationary measure. So a little comment on why you use the Henri's theorem. So since we're trying to deal with the probabilistic setting, so the idea is that we have microscopic data and we want to relate that with the macroscopic data. Is a really more natural setting when we start with a microscopic process. We're going to see, not in very much detail because it's fairly technical, that the proofs are constructive and the convergence is quantifiable. So it's not really decays, and in other hypocoercivity techniques using Krein-Rudman theorem or Poincaré inequalities. And it's also valid so for a broader class of initial condition. In particular, one that interests us is the Dirac delta. So we allowed ourselves to start with one single bacterium. There's a nice, really recent review by Hava Yoldash, which gives a really broad illustration of, in the unidimensional case, so not really similar to a setting, but to these hurried techniques to kinetic physics models and also to biological spirit inspired models. So the problem is that this Harris theorem I presented you was valid for the conservative case. So in our case that we have a non-conservative semigroup to sort out, we'll use the Joub H transform. So we'll create a conservative semigroup which depends in the existence of some eigenelements H and lambda for Q. So if I have the existence of a function H and a constant lambda such that I can solve this eigenvalue problem, I can define this semigroup P, which will be conservative. So I can apply then Harris theorem to P and then infer what happens in the long time with M. So in the model that I presented you here, it's really straightforward to see that a nice function H, and is indeed the only one, is the size. So H equals to Y, and the Malthusian parameters, so the population growth rate will be the elongation rate, so the unique elongation rate I established for the whole population. So in models when it's not very easy to find H and lambda, it might not be the best approach, and it would be nicer, I think, to use the non-conservative approach, and we'll see that maybe our constructions can all be useful in that kind of settings. So I will only give some ideas on the proof of the doubling condition, which is usually the harder one, because also the Lyapunov condition is really context dependent. So the idea is to start, so I have my PT, which is obtained using this transform here. So I can derive then the semigroup, the generator associated to PT, to P. And using that, I can prove this Duhamel formula, so that P is actually the mild solution of this iterative equation, where we have here this function phi is the deterministic flow, so it's my exponential growth, and this function kappa here gives the zero at each division for the H, and the continuous ratio given by my function P. So, and we will not consider so we have an equality here, so we'll consider only some trajectories given by that formula. So if you iterate that formula once, twice, you have what happens when you do one jump and two jumps and so on. So we'll truncate that at one jump. So that's what we have after one jump. So we see, for instance, here the probability. So this probability psi here is sort of the survival probability. So it's the probability of not jumping before this time t minus s. And what you have here, so is actually the value of f after doing maximum one jump and only one jump. So once we have here the idea to derivate the doubling condition, which requires to have a measure nu which is independent of my initial datum, is to do a change of variables. So I have here an integral that depends on z and s. And so if we are able to prove that this some C1 diffeomorphism that allows me to go from S, Z to A and Y, I can actually do the change of variables and try to minorate to try to find a uniform mu like that. So it's what you observe here, and to do that, we really need to use that we are allowed here to integrate in time. So to use, a measure mu that allows to take the average and time of the action of my semigroup, and using that, I can use the stochasticity of time to give another variable that allows me to do the change of variables. So since my function, so my measure mu using this change of variables is going to be explicit actually, so we can compute that for very different settings, and we have the function mu. So for different values, for different forms of f, so for the ratio between the two daughters, and we see beta function. The parameter of the beta function is the concentration. So the bigger the parameter, the bigger the concentrated. The concentrated is the distribution. So we see that if I tend towards a singular distribution when I have a delta in 1/2, my nu tends to be degenerate as well. And we actually know that in the case where we have exponential growth and a perfect f that is the delta in 1/2, we won't have convergence because we'll observe some periodic behavior. So it's what you observe here in a quantitative way, that the function nu that allows you to conclude the exponential ergodicity tends to be degenerate. The more degenerated is the distribution f. So really quickly, the idea, so in the other case, so in this particular setting of growing bacteria, we knew we knew h, we knew lambda, and we have some other explicit results that allow us to conclude. We can also try to do that in a more general setting. So I won't give all the technicities at this ongoing work that we hope to will be ready for you to read very soon. So when we have a general drift term, so a growth rate that is not necessarily exponential, and a jump dynamics that try to recover what you observe in the other. So we have a degenerated relationship, and so we have this kernel K that will be degenerate in a variable. So at each time you have a division, you have to come back to a deterministic value, as we observe here in the left figure, or to some deterministic function of your current rate, which can also be a source of lack of degeneracy. And the idea is that we can use the same idea, so use a UML formula, try to perform a check of variables using the fact that I'm allowed to include the time, so to average in time, to actually prove general doubling conditions in these kind of settings, which is not really trivial because we have the idea of the doubling condition is to explore the whole space, and here we're trying to explore the whole space using degenerate trajectories. So we go through curves in a two-dimensional space, and that's the main difference between our setting and the other Henri's theorem settings in the unidimensional case. So now really briefly, the idea is to use that now information to try to clarify what happens in real life. So we are actually able to observe the convergence in real life. So because we can observe the data in the Mother machine through time, so we took a picture of the cells at different spots in time, so we can follow the distribution here of the size, so of the current size of my independent cells through time. And we'll see sooner or later that my two histograms, so there are two different repeats, so two completely different initial conditions will fairly fast, converge to some unique distribution, of course, with some fluctuations around. What it was observed also in numerical interpretation in the right panel. So you have the difference between the current distribution and the mean distribution, and we observe that we tend to converge, and then we fluctuate around this mean distribution. And that's also a nice observation that using the mother machine data, we can observe actually not only the steady state, but also the fluctuations in time around that steady state. And one maybe perhaps of the next step in my thesis to explore that is to actually try to quantify these fluctuations in time around the steady state, which is something that we can only access using a probabilistic formulation like that, and it's really not explicit when you have an integral differential model. So we can do, of course, the same with simulations, and we can observe how the two compare. So really briefly, here we have the comparisons between the simulations in orange and the observed population in blue. So we have that there are some discrepancies, but we can receive fairly the same shape. And which is most surprising actually is that when we compare so blue and red are two completely different experiments. So the red one is the mother machine data. So it was observed when we do the average in time of my steady state, so of my sizes distribution using the whole data of the mother machine. And in blue is what we observe taking a picture in a petri dish. So it's two different experiments, of course, with the same bacteria, with the same medium. And we observe a pretty nice overlapping between the two, and it's actually allow us to formally conclude that what happens in the mother machine can be used actually to extrapolate to population, which is really not clear from a biological point of view. So I already discussed some of the perspectives already with you. So thank you very much.
Thank you for. Thank you for this enthusiastic talk. Are there any questions? Oh, thanks very much. Maybe a more statistical question. So do you intend to reconstruct B, I guess?
Yep.
So in a parametric way, a non-parametric, do you have ideas about how to reconstruct it?
Yeah, so for now it's parametric way. So if you have, since we have the distribution of the added size at division, since it's a parametric model, using that distribution, we can explicitly compute B, because we know mathematical ways. So we have the histogram of the added size at division, so we can fit a parametric model. And this distribution here is related to B actually, because this distribution here is usually so it's related to B in the sense that the integral of f, which is big F, is, as I said, is the exponential of minus B. So in some parametric cases, we're able to estimating F, so using this fit here to estimate B. There are also non-parametric ways to estimate B that can maybe explain why you have this not perfect fitting between what you observe in the population and in our simulation. Yeah.
Are there other questions on Zoom? No? So I think, yes.
Thank you. Yeah, I have a non-mathematical question, because I saw that you were working with biologists. So can you, what do you tell them that, like, I understood the general idea was to go from microscopic process to population, and can you summarize, like, in simple terms, what kind of results you end up telling them?
So first of all, we have that what we're saving, so in the mother machine, for example, you have the bacteria trapped. So it's not really clear to see that what the dynamics that you observe there are the same that we're going to observe in a population in a dish, for example. Because we have, so spatiality, for instance, in the microscopic setting, you don't have any spatial variable. So you have your bacteria that grows alone, and it's, for the biologists, it's not clear that what we observe in that experiment can give some ideas of what will be happening in a population experiment. So from a mathematical point of view, we give the justification of that, and we have quantitative ways to actually measure if we have at the end this overlapping of nodes. So here, for instance, when we observe this, when we have in the mother machine and in the bat population, and also in the simulations of the population models calibrated with individual data, where what we can conclude in this particular setting, because it's not a general truth, is that the microscopic data can be used to predict actually what will be happening in a population in a dish. So in this setting here, so we have also other settings when we have perturbed cells and it's not always true. And what we're doing now is to try to see why it's not true anymore. So, but using that, we can try to explore, knowing that in this setting it's true, why in other settings it will not longer be true. And this, I think, and I think that they think that as well is a really powerful insight on how to relate these two kinds of experiments.